Good afternoon, everyone, and welcome to today's webinar on food microbiology and lab water purification solutions by Merck. My name is Suha Afuni, and I am the Marketing and Training Manager at Gulf Scientific Corporation. So before we get started, I'd like to go over the format of this webinar, which will provide you with an idea about your participation throughout today's event. As you are already aware, your audio has been muted. However, you may send in your inquiries at any time during the presentation. We will be collecting your questions and we'll address them during the Q&A session at the end of each of the two sessions today. To submit your questions, please refer to the questions section in your control panel. Now, moving on to our webinar, I would like to talk a little bit about Gulf Scientific Corporation, GSC, and our partner Merck. Headquartered in Dubai, GSC was founded in 1990, so we're marking our 30th anniversary this year. With a team of over 200 professionals, GSC operates in over 10 countries across the Middle East. GSC has represented Merck for over 20 years throughout the region. And I would like now to introduce our panel speakers, Dr. Robert Nadaskay, Mr. Mohamed Hammad, Mr. Ahmed Werida, and Mr. Mohamed Elmallah. Thank you very much.
Good afternoon, ladies and gentlemen. My name is Robert Nadaskay, and I will conduct the biomonitoring presentation for you. Our agenda for today is as follows: single-pass and dual-pass products, hygiene monitoring in the food industry, tools for microbiology, Stericon Plus bioindicator, granulated culture media, Dilucult and Dilucult 2 and enrichment sample homogenizer, convenience Redibug dehydrated culture media, and the new color-coded MC media pads. As all of you very well know, the microbiology quality control is really a crucial point for the production of healthy and safe food and drinks. Of course, there are several areas of the microbiology quality control and also the hygiene control, which can heavily influence the final quality and safety of the food and beverages delivered to the customers. And therefore, the proper materials, methods, and procedures are very important for the high quality of microbiology QC outputs and results. And the internationally recognized methods and the guidelines should be used and fulfilled to prevent any mistakes and the false results. Especially currently, during the COVID-19 risk situation, is really necessary to keep the high quality and reliability of sanitization control and providing an accurate microbiology results of the food and beverages testing in order to prevent the contaminated food coming to the consumers. And of course, I would like to stress that Merck is a leading provider of the high quality products and solutions, being very well known as a manufacturer and supplier of various products for sanitization efficiency testing and food and beverages microbiology quality control. The first part of my presentation is related to the rapid methods for the pathogen screening in the food. Here we offer Merck immunoassay-based lateral flow test. We have the single-pass lateral flow test for one parameter, and we have a dual-pass lateral flow test for two parameters. In both tests, we have two zones. We have a control zone and the test zone. The control zone should always be positive. It means it proves that the test is working properly. When the test zone is positive, it means that the sample contains the respective pathogen. The workflow of application of lateral flow tests consists from the several steps. First, we have to perform the enrichment of the sample. Therefore, to the homogenization bag, first we add the enrichment medium, the broth. Then we weight the sample. Then we homogenize it. After homogenization, we incubate the sample. And finally, after the incubation, we pipette the sample and we place it on the lateral flow test. Then we wait 20 to 25 minutes for the development of the sample within the lateral flow test. And then we can read the result. You can see on the left-hand side the negative sample. In the negative sample, the control zone is always positive. There is a positive control, but the test zone is negative. There is no color. On the right-hand side, you can see the same test, but with the positive sample. It means here the both control zone and also the test zone are positive, showing the red color. So here we see the example of rapid detection of the Salmonella using the single-pass Salmonella lateral flow test. If we compare the lateral flow test against the traditional ELISA, we can see some advantages. First, the control reaction is built in the lateral flow test. It means that we don't need any separate reaction to demonstrate the proper function, which is necessary for ELISA, which also can save the costs and the money. Second, lateral flow test is done in one single step. After the application of the test sample to the sample port, there is no further pipetting steps necessary. And there is no separate color reaction needed for lateral flow test to make the reaction visible. And then also, lateral flow test provides the results faster than ELISA, and we need only time of 20 to 30 minutes for the development of the test, and then we can finally read the result. Currently, we have the following parameters within our Merck lateral flow test product portfolio. There is the single-pass Listeria, the single-pass Salmonella, the single-pass E. coli O157, and the single-pass of Campylobacter. And then also we have the dual-pass for the verotoxins and dual-pass for the Bacillus cereus enterotoxins testing. Next part is related to the hygiene monitoring in the food and beverage industry. It means there are the products for the testing of the efficiency of the sanitization for the food and the beverage production areas. The hygiene monitoring in the food and beverage industry is based on the concept called HACCP, which means Hazard Analysis of the Critical Control Points. The HACCP is a preventive safety system, not a reactive. It is a management tool used to protect the food supply against biological, chemical, and physical hazards. It is recognized internationally as an effective system of the food production controls. However, the HACCP is not a standalone program, and prior to its implementation, some prerequisite programs must be put in place. The typical of them are good manufacturing practice, good hygiene practice, sanitary standard operating procedures, then also the vendor certification and the recall and tray-back procedures. The important part of the HACCP implementation is the testing of the efficiency of the sanitization processes. For this are generally used the systems based on the ATP detection, adenosine triphosphate detection. For this purpose, we offer our product called Highlight system. The purpose of sanitization is to remove all residues of the biological materials from the areas of the production. The biological materials contain the ATP, adenosine triphosphate, and therefore we can use so-called bioluminescence reaction for detecting of the ATP residues. And then levels of ATP can be used to indicate the cleanliness of surfaces that could come into the contact with food, which we define as a critical control points. The relative light units measured by the luminometer are proportional to the amount of ATP. Measuring the light released by the reaction allows us to accurate determination of the quantity of the biological material. And our Highlight 2 is a luminometer which is counting the photons coming from the bioluminescence light reaction and giving us the value of the ATP present in the sample. Our Highlight 2 luminometer is a very robust instrument having specific features and benefits. First, it has a built-in light source, which is used for the automated instrument calibration on every startup. It means when you start the instrument, when you switch on the instrument, it automatically calibrates the photomultiplier against built-in light source. It means you don't need to have any separated calibration procedure or the calibration tools. Then the second benefit is the temperature compensation. The bioluminescent reaction depends on the temperature. It means different temperature gives different kinetics of the reaction and gives different results. The highlight luminometer is automatically compensating the result within the temperature range from 5 to 35 degrees. And when being outside of this range, the instruments gives you a warning message. The storage of the data is very secure because the data, the measured results, are stored in the non-volatile memory and will not be lost even if instruments loses the power. And if working in HACCP mode, data can only be deleted via PC. For collecting of samples and running the bioluminescence reaction, we have consumables called highlight pens. We have two formats of the pens. We have a surface test pen, which is coming with the swab and the rinse tube for the swabbing of the surface and extracting the sample into the rinse tube. And then we have a liquid test pen dedicated to be used for the testing of cleaning in place solutions for the presence of the ATP. These consumables are very resistant to chemical interference, providing dilution of the sample in the rinse tube and a high buffering capacity against the extreme pH. And also we offer the special application of ATP testing. There is so-called highlight jet fuel test. The purpose of that is to detect the biomass in the civil aviation fuel, especially in the big fuel tanks at the airports or in the storage facilities. The part of the highlight system is also software called Trend software. The purpose of this software is to help you to set up the limits for the critical control points, and also it gives you the graphical interpretation of the measured results. For example, here you can see the interpretation of the measured results within the time axis for the different critical control points, here marked as a depots. Beside of ATP-based highlight system, we also offer a simple tool for the cleaning monitoring called HiRISE. HiRISE is based on the detection of nicotinamide adenine dinucleotide. It is a color test strip based on the visual reading, so the no reader is necessary for that. It's a qualitative test, very simple to perform and giving the results in 5 minutes. All the reagents in the test are stable at the room temperature. And this product, this test, is suitable for the confirmation of the cleaning efficiency of the cleaning operations in various hotels, restaurants, catering, on the retail operations. It's very easy and simple to perform the test using the HiRISE test strips. First, you moisture the test area of the strip, you swab the surface, then you add the other reagents from the kit, you wait for 5 minutes, and you can see the results. If the test area remains without color change, it means the surface is clean. When the test area of the strips changes the color to the dark violet, it means that the surface is not clean and the cleaning needs to be repeated. We also offer several tools for your microbiology lab which can help you in your daily work. I would like to mention 2 of them. The first of them is so-called Merck anaerocooled system, which is dedicated for the cultivation of anaerobic and microaerophilic bacteria. It consists from the anaerobic jar or anaerobic bags and then the relevant reagents which create the anaerobic atmosphere in the jar or in the bag. So it helps you to rapidly establish or virtually oxygen-free conditions, provides the sufficient high level of the carbon dioxide. It's very easy to use, giving a high degree of the safety and reliable performance. It has also long-term stability and the problem-free storage of all components. And of course, economically, it helps you to save your costs. The second tool is so-called Stericon Plus bioindicator, dedicated for the testing of efficiency of autoclave sterilization. There are glass ampoules containing the spores of Geobacillus stearothermophilus, plus the growth media, plus the color indicator. So, when after autoclavation and which is followed by incubation of the Stericon ampoules, there is no growth of the Geobacillus, it means the sterilization in the autoclave was efficient enough. There is also no change of the color, and ampoules remain violet. If we see the change of the color from violet to yellow, it means that the spores of Geobacillus stearothermophilus are able to grow and change the color. It means the efficiency of autoclavation was not sufficient. So as mentioned, the Stericon ampoules are put inside to autoclave to check the functional efficiency. It means to test whether the prescribed temperature of 121 Celsius degrees is reached within the entire autoclave and whether the temperature remains constant over the whole prescribed period of 15 minutes. After this autoclave sterilization, the ampoules are removed and incubated for 48 hours at 60 degrees. And then we check if the color is changed or if color remains unchanged.
Thank you, Dr. Robert, for your presentation. Good afternoon, everyone. Just to introduce myself, my name is Mohamed Hammad. I'm working as a product manager for Merck Biomonitoring product at Gulf Scientific Corporation. I will start my presentation today with Granucult Media, which is a brand name of dehydrated culture media. However, Merck is certified as culture media manufacturer, various ISO norms, and has available a clear documentation about performance of the media. And we will see how Merck is compliant with ISO standard. As you can see here, ISO 11133 for microbiology of food animal feeding stuff. It consists of two parts. The first part for guidelines on preparation and production of culture media. Part two for practical guidelines on performance testing of culture media. This standard describes for the first time in the history of microbiology the definition of quality control for commercial culture media. We have a nice tools we will share it through WhatsApp and through email with you all to support you to find the part number of Merck. However, just to give some information about this one. So this brochure mentioned to you which part number you can use it and which media you can use it as per reference method you are following. For example, if you will say which media recommended for Listeria, if we will talk about DIN or AOC or BAM or ABHA. So it's recommended here to use triple sugar iron agar. So if you refer to this brochure, which I will share it with you later, you can find the Merck catalog number easily. And also we have a chromogenic media in our range for Salmonella, for E. coli, for Listeria mono, for Nakazaki, a lot. We have a lot of chromogenic media in our product range. Let me give one example for Chromocult coliform agar media. which used for simulation detection of total coliform and E. coli in drinking water and processed food sample within 24 hours. So after incubation time, you will see coliform with red color, E. coli with blue color, and even non-coliform you can detect it. To make summary of this dehydrated culture media, actually, our media is very compliant to ISO 11133, and our media is less dust creation during weighing, during the preparation in your lab, significantly reduce inhalation of toxic substance. As you know, most of media you are preparing every day consists of some toxic substance like sodium azide, selenite, lithium chloride, deoxychiclate, malachite green, sodium carbonate, and a lot. As you know, all this substance classified as a toxic, which can affect it badly to your human health, as well may cause environment pollution and also eye contamination, skin, uptake in the mouth. Don't forget that less dust equal to less risk of inhalation with dangerous toxic substance. Our media dissolve rapidly and uniformly when you prepare this one, and I'm sure all of you notice the difference between granules form, which is unique for Merck product, compared to powder form, and homogeneous distribution of media component during preparation. Once you prepare it, will be immediately homogeneous in your water. Most of antibiotics, it's included in our media, so no need to add any extra antibiotics. The shelf life of our media is 5 years from the date of manufacture. The storage condition will be at room temperature. And we have here in our warehouse most of media which common use in your lab. Please, if you want to ask about any offer, to contact our representative sales guy to check the offer. Here we have sample preparation and instrumentation. So actually, I will give you two examples: Dilucalt and Dilucalt 2, and enrichment sample homogenizer. So Dilucalt is automatic sample diluter, which is a gravimetric dilution, supporting you to weight the sample and added appropriate mass diluent. Add this liquid broth and buffer for your sample bag. Actually, this low profile can be put into a safety cabinet. It's easy to use and easy to clean, light cooled, different sample weight. So we can cover 5 kilo and we can cover 3 kilo. Our Dilucalt is adjustable parameter for pump selection, dilution factor, distribution mode, and speed. And even you can prepare 2 different bottles and have them on each side. So, for example, if you will testing every day a lot of sample for Salmonella and E. coli, so you can prepare both diluent or both cross media to keep it ready once you will start to work. After this one, you have to homogenize your product. So we have a nice product that's called ESH, Enrichment Sample Homogenizer. This is best to mix large variety of samples, especially hard and elastic like cheese, carrot, and pastries. This system is plug and play installation. It's very simple to use, and everything inside is stainless steel, easy cleaning, unique adjustable bottle system, variable speed, blending stress and time as well. It's reliable and low in maintenance because, as you can see, most of things inside, especially for bottle, is stainless steel and unbreakable window. As I mentioned, it's simple to use with digital display and easy to open and clean. Even easy to remove bubbles and reconnect it again after your work. Compact and robust and very long life time. For pathogen testing, we have a convenient solution, especially for Salmonella and Listeria testing, which can support you. Instead of preparing media by yourself, you can get our product. So we have our ready bagged media, which coming sterile. So we can cover especially two parameters for buffer Depton water and half freezer. So this ready bag, it's maximize your convenience during your workflow and minimize your effort as well. Pain point for you guys with large test volume of Salmonella and Listeria, I'm sure of that. So it's preparation and weighing of and autoclaving your media. As you know, if you want to prepare media to you every day, really it's taking long time. So if you will compare the time consuming between ready bag media and in case you will prepare the enrichment in your lab, then we can say your usual way, you have to weight your sample, then dissolve in water, you have to adjust pH, then you have to autoclave it, which really consuming at least 1.5 to 2 hours for autoclaving. Then you have to wait until your media becoming at room temperature. And if we consider all this one, really the ready bag is especially for high flow testing. So the benefit here, no weight of media, no autoclaving, no separate additive of supplement already included in our ready bag. Here, how to handle convenience ready bags for the hereditary culture media. First, you have to weigh 25 grams from your food sample in a sterile bag. Then you will add all contents of our ready bag sachet to this 25 grams of your product. After that, you will add 225 ml of sterile water. So we have our Elix system who can giving you this water, and I believe my colleagues Ahmed, Werida, and my colleagues Mohamed Elmallah will follow my presentation to give you more details about the water in your lab. Then after all this one, you have to mix by hand and place into our homogenizer, ESH homogenizer, sample to enrich both. Then you will incubate it and follow your procedure. Last but not least, we have a nice tool that's called the new color coded MC media bags. Our MC media bags ready to use culture media to support you in your lab. So it's convenient culture media for rapid enumeration of microbial contamination in food and beverages, compliant with international standard and quality control. International we have approved for ISO 11133 and Microval and AOC. So we have long shelf life as well. So shelf life will be from 2 to 3 years for our MC media bags. We have clear color coding for easy differentiation. So actually we can cover 5 parameters nowadays. We can cover E. coli and coliform and rapid bacterial count and yeast and mold, and soon we will have for Staphylococcus. If you will see the design of our MC media bag, you will see that every slide inside every MC media bag, you will see that it's color coding for different color, and inside you will see non-moving fibric pad for automatic diffusion of sample, and this will be saturated with chromogenic media to support you to get your result after incubation time. And in right and left, you will see visible guide for better readout, and also you can write the lot number, your sample details, the date of your sample. You can put all your data in this MC media bags. And each MC media bags, you will see it's have lot number and expiry date for this one. And just to say that our main objective of MC media bags only to support you to fill your gap between convention method and rapid result. However, we can cover a lot of your needs, whatever it's under dairy products, meat and fish products, processed food, and vegetable as well. Here you will see the procedure, how to handle this one. It's very simple, 4 steps. So first you have to open the cover fell from MC media bed, and after that you have to keep inside 1 ml from your sample. If you have liquid sample, immediately you can take 1 ml from your liquid sample and keep it in central of MC media bed. And after that, this 1 ml will diffusing inside our beds. If you have solid sample, as I mentioned, you have to homogenize first with your enrichment. Then you will take 1 ml from the mix and keep it inside MC media bed for some time. Then it will diffuse automatically. After this one, just to close the cover film. No need to push from top. Just close it, and after you close it, will diffuse automatically. And you can write your details about the batch number, the product, the date, everything. Then you can incubate it for some time. Actually, for yeast and mold, our bag can detect within 2 to 3 days. Really, it's very rapid. And also for total bacteria count, you can get your result within 24 hours. Same for E. coli and coliform. I hope you enjoy this art of microbiology with Merck Biomonitoring. And please don't hesitate to contact us if you need any more information about this product, and wish you all happy afternoon.
Thank you, Dr. Robert and Mr. Mohamed. We have received a number of questions, so we're just going to go over some of the questions that that our audience members have addressed to you. So the first question is, what is the shelf life of the ready bags?
Actually, shelf life for this MC media bag, it would be from 2 to 3 years. So I can say 2 years is minimum, 3 years is maximum.
Thank you for that. Another question that we received is, what is the sensitivity of duopath in terms of CFU?
Yes, this method is not so much sensitive, therefore it needs to perform the enrichment steps before. So the sensitivity is not so high. Enrichment is needed. I cannot tell you exactly which is the sensitivity, but I can share with you if you want. Yeah, but enrichment is mandatory to do.
Okay, and another question we received is, what is the pack size for MC media pads?
Actually, each part number for MC media bag consists of 100 pieces, each 25 coming separately. And once you will open this one, you have to close it within a month in the refrigerator.
Yes, in other words, there are 4 packs, each containing 25 MC media pads in the box. So together in the box there are 100 pieces.
Another question we received is, what's the advantage of the Stericon ampules over traditional autoclave strips?
Okay, yes, that's there are some other products to be used for the testing of the efficiency of the autoclavation. The advantage of the Stericon is that it's using the spores of this Geobacillus stearothermophilus, which is very sensitive for the keeping the right time and the right temperature. And yeah, there are the glass ampoules, so the product it's how to say, it's more sophisticated than the simple strips. So it's kind of the alternative using the spores, and afterwards using the incubation for the checking the color change. Yeah, this is the specific alternative for the testing of efficiency of autoclave sterilization.
Okay. Another question that we've just received is, what is the shelf life for Stericon bioindicator?
Shelf life, I am not sure. I am not able to tell you now, but I can share with you this information. with Gulf Scientific, and then they will send it to you later.
Yeah, just to say, shelf life for this one is 18 months from the date of manufacture.
Okay, we have a question from a member of the audience who's directing this to Dr. Robert. Could you please explain more about the highlight biomass test?
Yes, okay. It's very specific method. You know, usually in the tanks with the fuel, there are always certain part of the water. It's not so much, but there is some water in the tanks. And surprisingly, in this water are living the microorganisms. Okay, and these microorganisms living in this water fraction in the tanks can cause the corrosion. So that's the major problem, the corrosion. And therefore, in order to prevent this corrosion, according the regulations of the IATA, of the International Association of the Aviation, is mandatory to check the tanks with the fuel for the presence of microorganisms in the water. So, and these are highlight special test is dedicated for the extraction of this biomass from the water portion of the fuel, and then running the bioluminescence reaction for the detection of the ATP presence in these microorganism cells. So this is the principle. It's very specific application, and as I mentioned, based on the IATA regulations, the ATP testing is the method of the choice for the testing of the biomass in the fuel.
Thank you, Dr. Robert. We'll be taking 2 more questions, and then we need to move on to this next section. So another question that we've received is, can the ATP meter be used to detect allergen residue?
Allergen, basically no, because usually the allergens, as far as I know, there are the protein molecules, and the ATP is the molecule which is present in the biological cells. So the presence of ATP indicates the presence of any biological cell. So it's not directly related to the allergens.
And finally, is the rapid test suitable for honey samples to check the safety of consumption?
Sorry, I don't understand the question, honestly.
So is the rapid test suitable for honey samples to check safety for consumption purposes?
The honey. Okay, so I'm not sure which perspective of the safety of the honey you mean, but of course, if you want to check the microbial contamination in the honey, of course you can use the methods for the sample preparation which we have, and then you can detect the microorganisms in the honey. Yes, this is like with the other foods, so it's applicable. Yes, with the proper sample preparation.
Thank you very much, Dr. Roberts and Mr. Mohamed Hammad. We will now be proceeding to the next section of our presentation, which will focus on Merck's world-renowned MilliQ lab water purification solutions. Over to Mr. Ahmed Werida.
Hello, everyone. Following to the important information we received from our colleagues Rupert and Mohamed Hammad regarding the biomonitoring applications products from Merck, we are here today to speak about the importance of pure and ultra-pure water, which is the most common reagent used in different lab applications inside the industry of food and beverage. My name is Ahmed Werida. I'm the Merck Field Marketing Manager who is handling the area of Africa, Middle East, and Pakistan. I will be also supported by Mohamed Elmallah, who is the lab water product manager in Gulf Scientific Corporation, who is managing to handle the area of the Gulf region. Actually, water is the most common reagent used inside the lab. So whatever the applications you are handling inside the lab, water will have somehow interactions through direct and indirect effect. Applications such as dilution, sample preparation, washings, and final rinsing should be performed with highly purified water to avoid cross-contamination may affect the expected results out of the experiment. Plus, as well as we can consider also protecting the instruments we are working inside the lab. The agenda for today will be covering the basic informations you need to know about this magical liquid we are using inside the lab. And the topics we will be covering is water cycle and common types of contaminations, water purification techniques basics, highly purified water standards, norms and regulations, common water types linked to the applications we use impact of water contaminations on some lab application and on different instruments used inside the labs. The water cycle on Earth is how water continuously moves from the ground to the atmosphere and back again. As it moves through this cycle, it changes forms. Water is the only substance that naturally exists in three states on Earth: solid, liquid, and gas. Liquid states water will be found in surface sources like oceans, rivers, lakes, as well as underground. Accordingly, we will realize different types of contaminations that will be sticking with this water in both sources. Having a deep dive into types or the most common types of contaminations inside the water, the tap water we are receiving in our homes or laboratories will have 5 common types of contaminations. Ions such as cations, sodium, calcium, anions such as chlorine, sulfate, organics compounds such as artificial means, drug residues, herbicides, and naturally made like tannic acids and humic acids. Third types of contaminations are particles and colloids like sands, dust, silica. Fourth types are microorganisms and byproducts such as bacteria, endotoxins, protein enzymes, nucleases. And the fifth types are the dissolved gases like oxygen, carbon dioxide, and nitrogen. Accordingly, ultra-pure water is only H2O and nothing else. Several measurements have been considered to detect the level of contamination inside the water. In laboratories, we will be focusing on 5 types of them. The first one would be the inorganics ions. The measurements for it will be conductivity constant or resistivity, and the unit for that will be microsiemens for conductivity and mega ohm per centimeter for the resistivity. Organics measurements are the total oxidizable carbon TOC expression. The unit will be part per billion or picogram per liter. Particulates or colloids are measured by slit density index, fouling index. The units will be the rate of blockage of 0.45 micromembrane. Bacteria measurements will be the colony count on 0.45 micromembrane, and the unit will be colony forming units per milliliter. Endotoxins, the measurements is the rapid inculation test or LAL test. The unit will be endotoxins units per milliliter. In Merck Millipol, with 50 years of experience, we have developed a compact design for water purification techniques that is fitting with the limited space available inside the labs using the concept of disposable cartridges. The following diagram will describe the different levels of water treatments inside our water purification systems. Starting from the pre-treatment cartridge that is removing the contamination that can damage the system, such as colloids, particles, chlorine, and even silica, followed by the RO membrane reverse osmosis that is removing 95 to 99% of ion contaminations, giving type 3 water, and then followed by the electro-deionizer module that is offering us a consistent water quality not less than 5 mega ohm resistivity, followed by the UV lamp of 254 nanometers, which is killing bacteria, followed by the tank, which is a safe storing reservoir for the type 2 waters protected by a vent filter, to remove any air contaminations can be coming through the tank after dispensing the water from the tank. Also protected by an ASM UV with 254 nanometer wavelengths to protect against any bacterial growth inside the tank. The water then followed by a UV lamp of 185 nanometers and purification packs called Quanta, which is giving us type 1 water, which is removing any remaining contaminations of ions, and then followed by the dispenser of which is Q-put, that is also fitted with a 0.2 micron membrane to make sure that we are having type 1 water at the end point of the system. So what do we mean by pure water? In the market, you can see different names of the pure water, such as DI water, distilled water, ultra pure water, 18 mega ohm water, RO water even in some cases. But what is the real quality of these waters? It's not defined. Accordingly, we can consider these names as commercial names for pure and ultra pure water. In the next slide, we will describe exactly what is the water qualities we do need to use inside the lab. Laboratory water purification specifications have regulatory agencies which have published standards for defining the water qualities. Such as ASTM, the American Society for Testing and Materials, the NCCLS, the National Committee for Clinical Laboratories, which have developed to CLSI, which is the Clinical and Laboratory Standard Institute. Also, we have the CAB, which is the College of American Pathologists, ISO, the International Organizations for Standardization, the USP, United States Pharmacopoeia, EP, European Pharmacopoeia, CLRW, the Clinical Laboratory Reagents Water. All these agencies have defined the water qualities required for pure and ultra pure water definitions. In this table, we will be summarizing the definitions of each and every standards classifications announced by WHO in 2012. As you can see here, the ISO, the ASTM, the NCCLS, and Pharmacopoeia classifications. In ISO, they have defined 3 grades of water, grade 1, grade 2, grade 3. ASTM has defined 4 types of water, type 1, type 2, type 3, and type 4. NCCLS in 1988 have defined 3 types of water, type 1, type 2, and type 3, plus the Pharmacopoeia, European Pharmacopoeia, and USP has defined also several definitions of the water qualities required inside the lab to comply with this standard. In order to comply with the world standards norms for water, we have to define 3 types of water to be used inside our laboratory. Type 1 with the concept of resistivity higher than 18 mega ohm, type 2 with resistivity higher than 1 mega ohm, type 3 with higher than 0.0 mega ohm. Due to the cost, we need to estimate exactly how or what would be our consumption to type 1 water for critical applications only, type 2 for general application like buffers, pH solutions, microbiological culture media. For type 3 is for the glassware rinsing, heating passes, and feed to type 1 lab water systems. Let's have a general overview for the applications we use for type 3. It can be used for the autoclave feeds, environmental stability chambers, not silica sensitive, non-critical rinsing. For type 2 water, it is used for general laboratory applications such as titration, pH, UV, microbiology with 0.2 micron filter, glassware rinsings or final rinsing, and withering devices. Regular lab solutions such as buffers is very important to be used with type 2 water as it consumes large quantity of waters. Also, the glassware final washing should be used with type 2 water. Titrators, pH meters can also be worked with the type 2 water. Due to the cost consideration, type 1 water is the most expensive type of water, and it should be used only for critical applications like HPLC, LCMS, ICP-MS, cell culture, molecular biology reagents. And the rule of water in HPLC, for example, can be made for preparation of standards, mobile phases, washing, final rinsing of containers, blanks, and sample preparation. As a summary of the role of water in the laboratories, purified water can be used for plants, for dilution, dissolution extractions, final washing, rinsing for containers of analytical instruments, gel preparation, staining, preparation of standards, mobile phase for chromatography, buffer reagents preparations, bore motor passes, floating passes, tissue process, as well as feeding for lab equipments of autoclaves, glassware washings, climatic chambers. The water quality of pure and ultra-pure water are very sensitive to the atmosphere. Water usually working as a magnet to different types of contaminations may be existing inside the lab. So storing the water or the pure and ultra-pure water is not recommended at all. Using a freshly prepared ultra-pure water is the key solutions to avoid issues maybe happening during the experiments or the applications we are using the water for. Accordingly, my colleagues Mohamed and Elmallah will be reviewing in the next slides what would be the impact of contaminations and impurities existing inside the water you are using on the instrument, on the data interpretation, and even on the results you are expecting to get.
Hello, everybody. I am Mohamed Elmallah. I'm working as a product manager responsible for Gulf countries with the Gulf Scientific Corporation. So we will continue together to talk about the importance of pure and ultra-pure water in different lab applications. And now in the next few slides, we will see together the effect of water contaminants on the instruments, on the results, on the application. So let's see together what kind of effects that water contaminants can do in your lab. First of all, it can damage your instrument for sure. And if you are doing chromatography, it will reduce the lifetime of the column and extra cost will be added on you. Also, it may lead to loss of sample and reagent, erroneous results, and you may need to repeat the analysis, need to troubleshoot, and you will face difficulties in data interpretation. So there is a question we want to ask: how we think about the impact of water quality for each application? What is the potential impact of each contaminant on the results and on the instrument, and what are the possible implications for the analyst itself? In this slide, we will see the major components of an HPLC as example, and then to see the parts that can be affected because of water contaminants. So first of all, we have the solvent in the reservoir, and then the pump will take the solvent through the injector to pass through the column, and then finally to the detector. So where can problems occur exactly? For sure, the pump can be affected, the injector can be affected, the column can be damaged, and also the detector. In this slide, we will show some contaminants, and we'll see the effect of contaminants on your instrument itself. First contaminant we have the particles. Particles can cause severe damage to HPLC and UHPLC pumps and injector. It can block column and frets. It can increase the bed pressure as a result. Second contaminant is the bacteria. The bacteria can plug column and frets. It can increase the bed pressure as well. It can release organics resulting in ghost peaks. Third contaminant is the organics, which may lead to ghost peaks, baseline drift, poor repeatability, and interfere with the analyte measurement, and for sure will reduce the column lifetime. So the effect of particles and the bacteria on liquid chromatography, it will block the column and the inlet thread, resulting in high back pressure, leading to distorted peak shapes and poor repeatability. So the effect of total organic carbons. What is total organic carbons, or TOC? It represents the overall amount of carbon. It is an indicator for organic contamination in the water. It can be monitored online also. So in this graph, you will see the effect of the concentration of total organic carbon in the water you are using in the analysis. High total organic carbons resulting in ghost peaks, as you can see from the graph. So the storage of purified water will increase the risk of ghost peaks. In this graph, you will see the molecule water, which was stored in a plastic Nalgene carboy for a long time. On the other hand, you will see the fresh molecule water stored in a glass container. And you will see the difference yourself in the baseline. In the left side, you will see the ghost peaks, many ghost peaks you can see because of the storage of the water. As my colleague Ahmed explained earlier, that pure water it must be, or it should be, freshly prepared before the analysis itself. And that's what our molecule systems are providing to you. It's providing ultra pure water or deionized water on demand. So some problems and the consequences. The problems. The first one we have you may face poor peak resolution. So the consequences will be difficulties for quantification, and some peaks may be missed. Second, the problem is peaks with shifted retention time. The consequences may be difficulties in peak identification and data interpretation, and risk of error in peak identification. Third, the problem, unknown or ghost peaks, and the consequences may be lack of reproducibility, may be having implication on the production. Some problems are also like noisy baseline and drifting baselines. So for sure, there will be loss of sensitivity and lack of reproducibility. In all cases, loss of material samples you will be facing, loss of time for troubleshooting to repeat the analysis, loss of money to replace column and to repeat the analysis, loss of confidence and reputation in case of errors, delay in publication if you are working in academic sector. So the impact of environment on the water, on the baseline, or as you can see from the graph, you have two graphs. First one for freshly produced molecule water, and the second one for bottled water, which was stored for a long time. So you can see the difference between the two graphs. So it is highly recommended to use freshly produced ultra pure water or purified water. So the impact of water quality on the ionochromatography analysis. We have reviewed the HPLC in the last part, and now we will see the ionochromatography as a second example. So what ionochromatography consists of? First, you will see the eluent, then the pump, the guard column, the analytical column, the suppressor, and finally the detector. So when can problem occurs? So for sure, the problem can occur in the pump, in the column, in the guard column, in the analytical column, and in the suppressor as well. We will see also here the impact of water contaminants on ionochromatography. We will start with the particles, which may plug the threads, and also pollution and damage, may cause pollution and damage to the system, pump and column and suppressor and detector. Second, the contaminant is the ions, which may cause noisy baselines, shifty calibration curves, negative peaks, or reduced lifetime for the suppressor. Third, the contaminant is the organics, which may reduce the column lifetime, reduce the performance of the suppressor, may form complexes with the ion of interest. Fourth contaminant is the bacteria. Bacteria for sure may release bacterial byproducts like ions and organics, and will cause also the same effect like particles. So let's see the importance of purified water and the effect of contaminants on the microbiology analysis. So in the media preparation, the accurate preparation of culture media is one of the fundamental steps to ensure the integrity of microbiological examination. So since 99% of the culture media is water, So water quality is very critical when you are preparing your culture media. And the below specs should be observed when you are using the water. So the purified water you are using should be free of nutrients, should be free of toxic substances. The resistivity of the purified water should be more than 0.3 megaohms. It should be low concentration of chlorine, low concentration of heavy metals, and low microbial count. According to the international standards, ISO 11133, it also sets criteria and describes methods for the performance testing of culture media. It specifies the requirements for the preparation of culture media intended for the microbiological analysis of food, animal feed, and samples from the food or feed production environment, as well as all kinds of water intended for consumption or used in food production. And as mentioned earlier, the accurate preparation of culture media is one of the fundamental steps to ensure the integrity of microbiological examination, and it shall be given very special care. So here we will see some abnormalities when you are preparing your culture media and the reasons behind those abnormalities. First abnormality, you can see the incorrect pH. So the possible reasons may be overheating of medium during the preparation, or poor water quality you are using, extraneous chemical contamination, pH measured at incorrect temperature, pH meter incorrectly calibrated, poor quality dehydrated medium. The second abnormality is abnormal color of the prepared media, and this abnormal color can be obtained because of many possible reasons. First one, overheating the medium during the preparation, poor water quality, poor quality dehydrated medium, absence of one or more ingredients, incorrect ingredients use, incorrect pH, extraneous contamination. Third abnormality is the formation of precipitates, and this is maybe because of overheating the medium during the preparation, poor water quality, poor quality dehydrated medium, poor pH control. If preparing from individual components, so the impurities may be in raw material you are using. The fourth abnormality is medium inhibitory or low productivity. And this can be a result because of overheating the medium during preparation, or poor quality dehydrated culture medium, poor water quality, incorrect formulation used, toxic residues present in the preparation vessels or water, and control organisms incorrectly prepared. So you can see that poor water quality is a common reason to have sub-abnormalities in the prepared dehydrated culture media. So you have to give very special care to the purified water you are using when you are preparing your media. So what kind of water we should use when preparing our media? So first of all, tap water should not be used at all, because the contaminants may cause precipitation or inhibit the growth. So what kind of water should we use? We should use only the purified water. And this purified water should be free from compounds likely to inhibit or influence the growth of microorganisms, low in bacteria, and should be stored in container made from inert material. And the conductivity also should be more than 5 microsiemens. The conductivity of the water should be checked before use, and this is a very important point. So water which has been passed through ion exchanger can have very high microorganisms content. Take care. It is therefore advised not to use this process without checking the microbial content of the water itself. So highly contaminated demineralized water, even if sterilized by filtration, can still contain substances that are inhibitory for the growth of certain microorganisms. We have some other applications for purified water, like sterilization. So this media you have prepared should be autoclaved, as well as other solutions. So the pure water is required to produce very pure superheated steam. If the water is contaminated, so this contamination may cause some consequences and issues when you are preparing your medium. So hard water will form scale deposits on heating elements. Particles and metals may deposit inside the autoclave and on what is being autoclaved itself. Bacteria and endotoxins should be avoided. So finally, we as Merck providing a variety of products which can cover your application and even the types of water that you are looking for. So first of all, we have some water purification systems which produces type 3 RO water. like the Rios Essential, like the Rios system, and we have MilliQ HR7000. And this machine can produce up to 220 liters per hour of RO water. So according to the consumption you need per day, you can choose your system of interest. Second group of instruments, we have the type 2 water purification systems, which is Elix Advantage, Elix Reference, Elix Essential, Rios DI, and MilliDI, and the recently launched product MilliQ HR7000. And this machine can produce up to 150 liters per hour deionized water, and it can be connected in series. We can connect the 3 machines in series together to produce up to 450 liters per hour deionized water. We have very wide variety of products which can produce deionized water according to the daily consumption you need. And finally, we have the systems which produces type 1 ultra pure water, and it depends upon the feed water type. If you are feeding the system with tap water, so we have the MilliQ Integral, we have the MilliQ Direct, we have the Direct Q system. But if you have the source of purified water, then we have another group of instruments which produces type 1 water from purified water, like the recently launched MilliQ IQ7000, MilliQ Reference, QPod Element, Super Q, which can produce up to 12 liters per minute ultra pure water, and the Synergy and Simplicity systems. So this is an overview about the products we have to solve the issues and to provide you with purified water to be used in your laboratory according to your application. Thank you very much. And if there's any questions, it's more welcome. Thank you.
Thank you very much, Mr. Ahmed Werida and Mr. Mohamed Elmallah. So we've received a number of questions from the audience. The first question is it required to verify the conductivity of a product? Oh, excuse me. Is it required to verify conductivity of product water with a conductivity meter?
Actually, it is not required because we have resistivity cell before point of delivery of the water, and on the display you will see the value of total organic carbons, you will see the value of the conductivity or resistivity of the water itself. So no need at all to check again the water quality which is produced from our MilliQ systems.
Thank you very much. Another question is, how long can product water be stored without any changes to the conductivity?
Actually, it is not recommended at all to be storing the water, but if you have a big interest to store the water, it should be stored in a clean condition. For example, our tanks are equipped with a vent filter as well as a UV to protect any bacterial growth inside the container. Maximum stored water can be having freshly prepared quality at 1 hour. After 1 hour, it will not be any more ultra pure water, but you will be considering it as type 3 water.
Thank you very much. The next question is, for HPLC usage, is extra filtration for water required before applying it to the instrument?
No, it is not required because at the point of delivery, we have the millipack filter, which is 0.2 micron filter. So the water you are getting is free of from particles, free from bacteria, and with total organic carbons. I'm talking about ultra pure water with total organic carbons less than 5 parts per billion. So no need to refiltrate again the product water from the MilliQ.
Thank you very much. We're just going to take our last question because we're running over time. The question is, what is the difference between TDS and hardness of water?
So TDS represents the... Yes, Ahmed, go on.
Okay. Go ahead, Elmallah.
Yeah, TDS represents the whole ion contamination inside the water itself, but hardness refers to some specific ions like magnesium and calcium. And those specific ions resulting in scaling. In water which is having too much ions of magnesium and calcium, so we will call it hard water. But the TDS represents, as I said, all type of ion contamination inside the water.
Okay, well, thank you very much to all of our panelists and speakers, and thank you to our audience members for your time. We really appreciate that you've taken time out of your day today to be part of our webinar. As mentioned earlier, any questions that were asked, we will be sending a follow-up email with answers to your questions. And if there's anything that we can do, please do get in touch. You can just respond to the confirmation email, or you can contact us on marketing@gulf-scientific.com. So for now, thank you very much. Be well, be safe, and all the best.